So good morning, everybody. So for those of you who don't know me, I'm Kate Lewis, and I'm one of the faculty co-directors of Women in Science and Engineering, or WISE, and also a professor and chair in biology. So I want to welcome everybody to our 19th annual Norma Slepecky Undergraduate Research Prize Awards and Memorial Lecture. Dr. Norma Slepecky was a greatly respected colleague, a distinguished auditory neuroanatomist, and a professor here at Syracuse University. Each year, we celebrate her commitment to the inclusion of women in science and engineering and to undergraduate research. The legacy of Norma, who was a professor in biomedical engineering and neuroscience, and also of her husband, Ralph, who was a professor in biology, and her family can be found throughout this campus, but particularly in this event. We are really pleased to welcome today Norma and Ralph's daughter, Anne Slepecky, and her daughter, Charlotte, who are joining us today. Thank you, Anne. Before continuing, I would like to offer our traditional acknowledgement with respect for the Onondaga Nation, firekeepers of the Haudenosaunee, the indigenous people on whose ancestral lands Syracuse University now stands. Honoring the contributions of women in STEM and encouraging undergraduate women to be active participants in research are important parts of building a new and a more inclusive future. The mission of WISE for over 22 years now has been to foster an inclusive STEM environment by supporting the recruitment, retention, and advancement of women in STEM at all levels of the pipeline. WISE achieves its mission and the mission of Syracuse University by continuing our Vanguard programs for first-year students through faculty, and by carrying on the legacy of Drs. Norma and Ralph Slepecky, who are both members of our faculty who are committed to inclusive excellence. WISE, in all that we do, is led by women STEM faculty, such as Norma Slepecky, who was an original member. Their leadership has directly impacted the status of women in STEM at the university for over 22 years. However, we could not do this without the many faculty, both women and men, who volunteer their time to support women undergraduate students and WISE. At this time, I would like to introduce the Norma Slepecky Undergraduate Research Prize. So today we recognize and celebrate independent research conducted by our undergraduate women students across the campus STEM departments. The Slepecky Prize is presented annually to students who demonstrate excellence in scholarship and who submit the best article, thesis, manuscript, or presentation from a professional meeting in the sciences or engineering. The award is made possible by the friends, family, and colleagues of the late Dr. Norma Slepecky, who endorsed the endowment of this prize. In reading about her work and her legacy, I was struck by her commitment to her students and their trajectories. I was noted by the Association for Research in Otolaryngology, a society for the medical specialty focusing on the ear, nose, and throat. Norma attended her last doctoral student's defense just 2 weeks before her death. Her devotion to students and to women in STEM fields in particular is to be celebrated. We also hope that it will be emulated by generations of scientists to come. Before I announce the awards, I'd like to acknowledge the contributions of several people who have helped make this competition possible. First, our distinguished reviewers. The committee included faculty from the departments of biology, civil and environmental engineering, earth sciences, electrical engineering and computer science, physics, and psychology. Their task was especially difficult this year because the quality of submissions was uniformly exceptional. We appreciate their enduring support and service. Please join me in thanking these faculty for their service with a warm, silent applause. And I invite you to use the sign language for applause throughout this seminar, given that we're on Zoom. I'd also like to acknowledge the faculty mentors who have encouraged their students to be actively involved in research as undergraduates here at SU. The submissions we received for this competition make it clear that doing so is an important and a worthwhile endeavor. I would invite you to again give a round of applause for all of the faculty advisors who make these opportunities available. Lastly, I'd like to thank the applicants for the privilege of reading their work. The reviewers were repeatedly impressed at the sophistication of the research and the clarity of writing of the applicants for this award. This competition demonstrates the vibrancy of independent scholarly research by undergraduate students at this university. We wish we could give awards to them all. This year we have 2 awardees for this competition, first and second place. Zoom has been proven to be a remarkable platform during this past year, but we know that with this medium, it is more difficult to duly recognize awardees and their nominators' advisors. So what I'm going to do is to ask that when I name the awardee and the advisor, that they unmute themselves and say something so they will show up on your screen and you can see who they are. So our first place awardee is Laurel White. So if Laurel could say something.
Hi, I want to thank everybody involved with the competition, as well as my research group and my department, and I'm very honored to have my research recognized like this.
Wonderful. So Lauren is mentored by Dr. Duncan Brown. Unfortunately, he had a previously scheduled NSF review panel commitment, so he's unable to be here today. But he asked us to offer his congratulations to Laurel and his apologies for his absence. Laurel was unanimously selected as the first prize winner of this award, given her outstanding work in the area of physics. The title of her reviewed honors thesis is The Effect of Spin Priors on the Determination of the Neutron Star Equation of State using Gravitational Wave Signals. This project focuses on challenges with the parameter estimation used to analyze gravitational wave signals. Her nominator, Dr. Brown, notes that the work Laurel has done has led to the significant discovery that it will be more difficult than previously thought to get an accurate measure of the equation of state with current second-generation detectors, due to the challenges of extracting the tidal deformability from the waveform and the impact of prior assumptions on the neutron star mass and spin distribution. It is expected that this research will lead to 2 articles before she graduates this year, adding to the list of already completed publications. Please join me in raising your hands and applauding Laurel. So our second place awardee is Aliza Wilsey, a senior in aerospace engineering. Aliza, if you'd like to say something.
Hi, I want to say thank you to the selection committee for selecting me, and also congratulations to Laurel.
Thank you. And she was nominated by Dr. Youngmin On. So if you would say something.
Oh, thank you. And then first off, I'd like to congratulate Eliza on this well-deserved award. And Eliza has a truly amazing with the way she pursued her goals with the passion. And then I look forward to continuing to watch her grow as a PhD student, since she officially got admitted to our PhD program. And then I'm confident she will continue to achieve greater and greater things.
Wonderful, thank you. So for the nomination submittal, Aliza provided an ASME conference proceeding for which she was the lead author. It is titled Investigation of Mycelium Growth Network as a Thermal Transpiration Membrane for Thermal Transpiration-Based Pumping and Power Generation. She is continuing to explore in her research the benefits and results of the mycelium membrane, with an additional paper expected. So congratulations, Aliza, on your award. And again, please can we raise our hands in applause? May we have one more round of applause, please, for both of the 2021 Norma Slepecky Undergraduate Research Prize awardees before we move on to the lecture. I would now like to introduce Dr. Shobha Bhatia, who is the other faculty co-director of WISE and Emeritus Professor of Civil and Environmental Engineering. Also a recent awardee, I would like to first congratulate Shobha on her recent Seinfeld Scholar Award. She will introduce our Slepecky Memorial Lecturer.
Thank you, Kate. Before we start, I would like to thank Humanities Center and the Department of Biology for their support for this memorial lecture. For many people, art and science and engineering have been considered 2 separate disciplines, but for me, the work is synergistically. A great deal of creativity is required to make scientific breakthrough, and art is just an often expression of or based on scientific knowledge. I've encouraged my student to draw or build a model instead of writing a paper. And students are initially surprised and hesitant, but generally are very happy with the outcome. Our speaker today, Dr. Ahna Skop, brings the life to the synergy between art and science. Dr. Ahna Skop, as an associate professor and geneticist at University of Wisconsin-Madison Department of Genetics. She is also affiliated with Life Sciences Communication Department and UW-Madison Art Institute. We are proud to say that she received her Bachelor of Science in Biology and Ceramics from Syracuse University, class of 1994. She went to receive a PhD in cell and molecular biology from University of Wisconsin-Madison and conducted her postdoctoral work at University of California, Berkeley. The junction of being a scientist and artist is evident throughout her education and professional career. Her lab seeks to understand the molecular mechanism that underlie cell division during embryonic development using Nemodate C. elegans as a model system. Failure in cell division often lead to birth defects, cancer, and age-related neurodegenerative diseases. Understanding how cell divides is highly dependent on vivo microscopy and large amount of visual data, which dovetail perfectly with one of her other passion, art. The combination of scientist and artist inspire her to think differently and maintain an open mind. Some of her work can be seen in the main entrance of Genetics Biotechnology Center on the UW Madison campus with 40 feet scientific art piece called Genetic Reflection. She also curated and contributed to a traveling exhibition of scientific art called Tiny Art from the Microscope and from the University of Madison campus, and she organized biannual warm art show for the International C. Elegant meeting for 24 years. Her science and art has been featured by Apple, The Scientist, USA Today, Smithsonian, PBS.org, NPR, and Science magazines. She mentors both scientists and art students in her lab and serves on the board of Wisconsin Science Museum, where many of her art science collaborations are on display. Dr. Skop has multiple publication, presentation, and art installation. She has also received prestigious Presidential Early Career Award for Scientists and Engineers in 2006 and named as a remarkable woman in science from the American Association for Advancement for Science in 2008. In 2015, she was honored as a Calvi Fellow from the National Academy of Sciences. In 2018, she was awarded the first Inclusive Excellence Award by the American Society for Cell Biology and Howard Hughes Medical Institute. She has also been recently named STEM Superstar at UW Madison by the Breva magazine. Finally, we wish to note that Dr. Skop is a fellow, WISE, Women Advocating for Inclusion of Women and Students of Hispanic, Chicano, and Native American backgrounds in STEM. Her work reflects her multiple identities as a woman, scientist, artist, food blogger, and author, as well as her artistic upbringing, her Eastern brand, Cherokee, Ukrainian, and Lebanese family heritage. Please join me in welcoming Dr. Ahna R. Skop, class of 1994, who will be giving a talk titled Too Creative for Science.
Thank you so much, Dr. Batia. That was I'm very honored and embarrassed. I feel like sometimes amazed I have accomplished a lot of things since I left Syracuse. But thank you so much for having me here. I am going to later in my talk. So for those of you who like to fall asleep, you can fall asleep. But later on, I'm going to quiz you, and it's going to be very interactive. I'm going to have you guess things on my screen. And I want you to unmute yourself, and you can kind of shout out. So, but before I begin, I'm honored to give this lecture in honor of Dr. Norma Slepecky. I knew her before she was a faculty member. I took a class with Ralph Slepecky when I was there as an undergraduate, but I knew her. But I'm honored to give this talk because it's near and dear to my heart, because I'm passionate about undergraduate science education, and also in making sure that there are more women in science. And so I'm honored to give this memorial lecture in honor of this amazing, remarkable scientist. So the title of my talk is called Too Creative for Science. There's supposed to be a question mark at the end of that talk. This is a story of my life. So this is a path of my life. So when I was in graduate school, one of my first mentors, I joined a lab. Eventually I left that lab, but he told me I was too creative for science. This was the second year of my graduate school here at Madison. Needless to say, I was devastated, and I left lab for about 2 days. And so I'm going to tell you what happened after that. Obviously, I'm still here, but I'm going to tell you what happened after that. So this was part of a project I'm showing you. We were coming up with a ABC book about C. elegans, and this is E. coli printed on Petri plates that our nematodes eat. So this is this too creative for science idea. Am I too creative? Is it possible to be too creative for science? So I'm going to weave you through my path in life. So I'm going to tell you a little bit about my family, about being too creative, science, and then science as art. So as a geneticist, before I begin, the most likely place to start is to share you a little bit about my genetics. And most of you probably have seen this before. Everyone's probably seen a pedigree. So I'm going to walk you through my pedigree. So my father, who is a Syracuse grad, played football in the 1950s, was a sculptor, medical illustrator, and anatomy educator. He's of Ukrainian descent. My mother, who is on this call, is a ceramist and an art educator, and she's Lebanese and Eastern Band Cherokee. And here's a picture I recently found of my mother and I in 1972 when I was born. My mom was my dad's grad student and went to Southern Connecticut. My dad taught at Southern Connecticut and also at Yale at night as a sculptor. They got together, obviously, and have 4 kids. I'm the oldest of 4. And the first thing that you're kind of looking at now is like, my gosh, Ahna inherited the recessive gene for science. Because if you know anything about genetics, right, there's only one scientist in this family. My dad kind of did medical illustration. My sisters are both amazing graphic designers and marketing execs, and my brother's an industrial designer. So these last 2 also are Syracuse grads, and my sister and brother worked in the biology department for a short period of time as well. And a lot of students are kind of wondering, well, maybe the doctor in the family makes the most money. It's actually not true. I make the least amount of money because art and graphic design is a very important business, as many people know. And so a lot of people ask me, well, how did you end up a scientist? And I always like to tell a joke is that because my dad's name was Microscope, that I became a scientist. So my dad was Michael Skop, and his high school biology teacher, he didn't have a middle name. His high school biology teacher says, you have this fantastic name. You should actually have the middle name Rowe. So my dad legally changed it to Rowe, and he drew a microscope at the bottom of all of his drawings. So microscopy has been part of my life since a childhood. I just didn't know that I would end up a microscopist, but it's kind of odd because that's what my whole life is about. It's kind of like my dad and the microscopes are kind of intertwined. So this is my genetics. So this is a picture. This was in the article that came out at SU this week. This is my dad in my basement of my home doing the bust of Ben Schwartzwalder, who was a very famous football coach at Syracuse, who was a coach of Ernie Davis, who came after my dad. My dad was the center for Jim Brown, which all of you probably know. My dad was drafted to the Steelers, but didn't go to the Steelers, but a lot of the teammates at that point did go to the NFL. My dad decided to go on a Fulbright to Europe. So he decided art was more important than doing football. Sorry, but this is my home, right? Nothing about this is sterile, right? This is a very dirty home, and my mom's on here. This was the bane of her existence. So having clay and plaster and all of these things around our home. But this was my environment in which I grew up. This is all I knew. I didn't know any different from this. My mother, this is one of my favorite paintings of my mother's. My mother is a ceramist, however. Here's just a picture of one of her plates. But was a grad student of my father's, but she is this amazing art teacher who's now since retired. She does anything from fabrics to jewelry and enameling, all these things. So my house was just full of lots of art. My father had an art school at our home. It was a private art school. People came from all over the world. Here's me at 2 years old, and here's my mother. My dad's not in this picture. This is 1974, and all of these people, of course, this is the 70s, there's a bunch of hippies, you know, this is my environment. A lot of people are still professional artists today. She's an actress in New York. He owns a gallery in Los Angeles. She's a famous ceramicist and a chair of a department, and there's lots of artists in here. I thought these were my brothers and sisters, but they were kind of students I grew up with in my home. I found the beauty of social media finds pictures of yourself that you don't know exist, but this was a picture of me and my dad's favorite chair. Again, a very dirty chair, another bane of my mother's existence, but my dad loved this chair. And this is 1975. If anyone knows me, knows anything about me, this is the last time I was serious. So 1975, that's the most serious you see my face. And you'll know this because in 1976, I'm here and I'm kind of coming into my own personality. Here's a picture of my dad. And then my two sisters are here. My brother's not born. Here's my mother. Again, amazing, remarkable photos. So these were kind of family photos with the artists that were in our home, and they all still impact my life immensely. But I grew up in this very rich, dynamic environment. Being around these people from everywhere, giving me ideas. There are even archaeologists that came, musicians. It was just a really remarkable time and a very unique childhood. So the other unique thing was my father had answered an ad in the newspaper. We lived in Connecticut at the time, and we won this home. So we had about 2 kids at that time. My dad was making $11,000 a year as a faculty member in the 70s. My dad answered this ad and we won this home because my dad wanted to always have land and have a studio. And Julian Bechtold is a famous artist in the Cincinnati area. And probably you might know what he did, but what happened in this home is quite remarkable in the history of combining art and science. So Julian Bechtold actually worked for Procter & Gamble and carved the first ivory soap bar, which was done in my home. And this was very apparent to me that my dad would tell me, he's like, well, the chemists kind of make the soap and they know how to press it. The engineers would know how to press the soap, but you need art to sell this. You need to have the design of that. And so I knew that from very young that art and science had to come together to make things work. And so this actually happened in my home. And so this was Julian Bechtold. And so the only stipulations of winning this home is we ended up living with his sister that never married. And so she became our grandmother. So I think nowadays where people pay mortgages, all our stipulation is to kind of have a new grandmother. That was kind of a good payment for a house. And so we ended up living there and my dad could have his art school. So another amazing, remarkable opportunity of being in a very special place like this. This is the back of the home. My dad had learned we would have gardens and my dad built this for my mother. This is a rose garden. And we had sculptures everywhere. So it's odd for the outside world, but for me, I just thought this was totally normal. And when I left for when I left for Syracuse, my dad pulled me out to this rose garden, and there's actually quotes that go around this. And if anyone knows me, I really, really love quotes, and they're very inspiring. My dad brought me out to one of these squares before I left for Syracuse, and he said, he said, every child is an artist. The problem is how to remain an artist when we grow up. So this is a Picasso quote. What my dad was telling me, and I didn't realize at that time, is don't forget who you are. Don't check yourself when you leave for college and forget this amazing environment, right? And I think for all students out there, you think that there's a way to do science and a way to be in science. But my dad instilled in me this amazing quote that kind of drove me. It's like, I am still an artist, and I always will be an artist because genetically I am an artist, right? But don't forget that, right? So I left for Syracuse, and I found this picture in Norway, which kind of describes, I think, my own life, but I think probably a lot of people's lives. Life is a long, windy path full of very treacherous roads to get over. And so I left home. I left this very rich, dynamic, artistic home, and I ended up at Syracuse. I started out in biology, and I also minored in ceramics. Margie Hutto is still a faculty in ceramics, and I was a student of hers. And I recently saw her at a conference last month. And so I got my degree in biology and always was in the studio with clay, coming into the biology labs, doing my molecular biology. I then was inspired to go to graduate school by Eleanor Main, who was in the biology department. So she had been to Madison. The last place I applied was Madison, and now I'm a faculty. And this, I will tell you a story. This is where my too creative story comes up. This was kind of like the bridge that I had to go over because it was a very difficult time in my life. And I thought I was going to leave science, but I'm still here, obviously. So the story is a positive one. And I end up going to Berkeley and to do my postdoc. And now I'm back here as a faculty in Madison. So here, just to convince you, I graduated at Syracuse. This is my motley group of roommates. We were both biology majors. She has, my roommate Denise, what has her chromosomes on her cap. I had DNA, and here I am. I just found this yesterday. So Jeremy's mom found it. They were poli-sci majors and president of student body and vice president. And Laura actually ended up being sort of a CEO at Yahoo, and they both are kind of poli-sci, and Denise became a scientist as well. So this was me at Syracuse. I left and I went to Madison. And the first 2 years of graduate school were a very difficult one for me because even though Syracuse was very challenging and hard, I didn't realize that my entire class in graduate school would probably be they would be valedictorians of their class at every college around the world. So that was actually kind of, I was shocked, but I ended up joining a lab, and I had this mentor at the time who said, "You're too creative for science." And I was devastated, right? So how do you think that might make me feel after you've just heard my story, right? Really even more devastated, right? But this mentor did not know any of my background, right? So this is the importance of being a mentor and understanding there's more to a person than just paper or other things. There's really a rich dynamic background there and culture and all of these things there. So I called my dad, this is before cell phones, and I said, "Dad, this happened." He said, there's two assholes on every campus and you found one. This is how my dad used humor with this. And he says, "Go to the library and I'm going to show you that you belong in science." This is what my dad said. So I did. And we went to the old stacks. And of course, who do I discover? Right? Leonardo da Vinci, right? So my dad got there, and this is back when science were artists. So my dad walked me through the fact that artists were always there. And my dad had my mom had an interest in science as well. And so that phone call kind of changed my tune about staying in science because I really didn't feel welcome at that time. I then found other scientists that I also were artists that I didn't really realize. This was a remarkable, drawings that I found from the scientist Ernst Haeckel. who actually was involved. If you Google him, you'll find these remarkable images of mushrooms and diatoms. And I just was like, wow, like I just didn't know. I never was taught this in high school. I didn't know this in college. I was just overwhelmed with this. Strangely, he looks a lot like my dad. So I was like, okay, you know, there was kind of a friendly face there popping up. Ernst Haeckel was involved in communicating to the public Darwin's theory of evolution through drawing, right? So I knew science comm was happening way back then, and Ernst Haeckel was part of that. So that's kind of a cool story. The other person I discovered was Ramon Cajal, the father of neuroscience. These beautiful Purkinje neurons, as you may have seen, there was a recent show that has been traveling the world from the Cajal Museum. If you saw it, I actually flew to New York City just to see it because I was always wanted to see his drawings in person. He was an avid painter and artist, but his father said, you need to go to medical school, right? And so he did, but actually these were the talents to his success. So I'm discovering all of these amazing scientists that had art was a real key to their success. And when I was at Syracuse at that time, there were only 2 women faculty members that I knew. And I'd never met that many women scientists in my life, and I never really knew the history of that. And then I discovered, obviously, Rosalind Franklin, that many people know, and her amazing X-ray diffraction image. And it was at that point in my career, I realized that after I did this soul searching is that there is really an importance of visualization in the discovery of science. And if it wasn't for Rosalind Franklin, as you know, these 2 guys, Watson and Crick, couldn't have built their sculpture, right? So all of these things, I saw that art and science really were intertwined and really important. We in science call these sculptures models, right? But they're sculptures. These armatures, a lot of them come from the art world. And so scientists took these things from art, but certainly used new words. And over the course of my career, I would look for quotes that kind of kept me going in my life. And I found this quote later in life, but I put it in this point in my life is this psychologist at Berkeley, Alison Gopnik, said, scientists are actually the few people who as adults who have protective time when they can just explore, play, and figure out what the world is like. And she says, scientists are simply big children. And at that point, I realized I'm kind of a big kid too, and I love science and it's kind of fun. So I realized I should stay in science, right? So that Too Creative for Science comment, even though it destroyed me, it allowed me to do a little soul searching and decide, do I really belong here? And then I realized I do, because look at all these famous scientists. And thank the Lord that my dad had that conversation with me to get me to the Da Vinci connection, right? And so that was really a really key part of my life. So in graduate school, I had also lots of other difficulties that I like to share publicly. I failed my prelim twice, and most people who have a PhD know this is kind of a big deal, and usually you're kicked out of school at that time. But I had one, the best mentor ever, John White, who came and said to me, and I was really distraught, and I realized all my classmates thought I was an idiot, and he said to me, he said, he invented this microscope. He's like, I got a D in math, but I invented this microscope. That was another transformative quote that was said to me. I looked at him and I said, are you kidding me? You made this microscope? You have to know a lot of math. And I'm horrible at math because I'm a dyslexic. And turns out he also was a dyslexic. And this was the new mentor that I had in my life. So I switched labs from the other lab, and I realized that this person really got me. He not only loved art, was dyslexic, and he had failed other classes as well, and he was very successful. He's also he was the first person to map the nervous system, and still to this day in C. elegans, it's the entire nervous system, each nerve that is born in the worm. So I was honored to be part of his lab, and if I didn't make that change and find this mentor, I wouldn't be in science today. So the other thing that happened in grad school was I found out I was in an RNA lab running a lot of gels. People have seen these kinds of DNA gels. I didn't like these things. I was like, is this really biology and science? I kind of was like, I'm not sure. Like, as a dyslexic and a visual learner, I was like, I don't know what I'm looking at. Like, it was very hard for me to understand. And I was like, I don't really like this, but I found out that people like to put this as art in their house. They send their genome off and they put it up in their house. I could care less about DNA gels. I still don't like them. I find them very hard to see. But what I discovered is I fell in love with this. My mentor was involved in video microscopy too. So this is one of my first movies I did on the multi-photon microscope that he also worked on. This is DNA aligning. This is sperm and egg have met and the mitotic spindle sets up. I can look at this time and time again, and I absolutely love it. Lots of people love mitosis, but I fell in love with this. And I initially fell in love with actually the static image from Eleanor Main's lab. So I took her development course and I actually circled this embryo in the textbook that I had at Syracuse. And I ended up meeting the scientist who did the microscopic image there. It kind of was fortuitous, but I fell in love with this. And it dawned on me why I ended up liking this is because my actual favorite artist is Moreau. And so what you could notice is there's a nucleus and these kind of asters and the microtubules there. Moreau is a very biological artist to me, but it dawned on me like these two things were intertwined. And it was like, I wouldn't be a cell biologist if I didn't kind of love this kind of art. And so I was drawn to like trying to figure out science by the beauty of it first. And it was because of my background in the arts, certainly, and finding different artists, and Picasso, certainly I love. So I still work on this today. I'm still very passionate about the process of mitosis. I'm not going to tell you the details of this, but if you come to my talk on Monday, I'm going to be talking about this structure that everyone thought was a garbage can, which is this little pinch point of the cell called the midbody. And it happens right before the cells separate. And this is a beautiful event that continually fuels my artistic side of myself. And every time I look at it, I see something different, and I'm always excited to see it. And to see it live it's like a painting come to life. It really is a remarkable thing to watch, and just an honor to be able to see it. So I'm very proud of a lot of things in science. I've done a lot of things in my life. So I'm a cover girl of science, sort of how I got my job from my postdoc. The other things I'm proud of, I'm in the laboratory of genetics at UW-Madison. And the reason why I'm proud of this is that in 1994, I was denied from this department as a graduate student. And why? Because my GRE scores were low. I was one of those people many people lost to the graduate school because my GRE scores are low. Now people are obviously changing this. There was a scientist called my professor at Syracuse and said, she has the GRE scores of an artist. And he said, well, she is an artist. So it was kind of like this thing. And I decided that I fortunately got into cell molecular biology program, but I got rejected from my department. But in 2004, I became an assistant. I was associate in 2000, and I'm now a full professor in 2018. So I'm very proud of being rejected in the department of which I am. The irony is not lost on me. And I think that's also an important story is that you're not going to get into all your things, but you can still be here. You know, this is not about your intellect. It's about this. It's about persistence, right? Persistence is really important. And I love this quote is like, don't judge me by all of these things, the science covers, the things I'm doing, but how many times I got up again, right? And I think for students, you're always going to have some failures in your life, and it kind of knocks you down a little bit. And if you still like it, you keep going, right? And so persistence is really part of my life. And I'd say that's really where I always am. I like this, so I'm going to stick this out. So to switch gears a little bit, so what happened after? So I get a PhD, I get a postdoc, I become a faculty. And then during my graduate school, I became very aware that all these images I'm seeing, and I'd done art shows with my mom and dad. Why the heck, with all this taxpayer money, I grew up in a low income home. We had all these art shows, but I'm doing the science that I know my taxes are going to that weren't being shared with the public. I'm like, why the hell don't we share this stuff? It didn't make any sense to me because it's so beautiful, because I just was busting at my seams to share it, but there was no mechanism to do that. So grad school, I started doing drawing. So I did logos for the meeting. I've been doing it every year. I've never paid to actually go to a conference because I always do the logos, so it saves my lab money. But I always do these, but it was kind of my outlet of like, how do I communicate a really beautiful organism in a different way through the art? So this is probably the most famous one. This was one of my first ones I did. And then from that became the C. elegans art show, and which I still do today. I'm doing one this summer again. So in 97 as a grad student, I asked my mentor, you know, can I do this show? He says, you can do it, but don't include me. So John White says, I'm too busy, don't include me in this. You do it yourself. So as a grad student, I did the first art show. And what happened was my postdoc in my lab says, Ahna, you're doing a mistake. Your career is going to be over. And I looked at him, I'm like, wow, I don't give a shit. If I do it anyway, I want to do it because it's still there. Obviously I'm still here. And this person, you know, and you're always going to get hit down by people that don't understand. They think there's a process to become a scientist. And I realized I'm just going to do it because I want to do it, right? And so guess what happened from it? It's still an ongoing event, right? I got lots of press in Science, the Scientists, and lots of Genetic Society newsletters. And then when Sidney Brenner, the Nobel laureate Sidney Brenner, who started the C. elegans field, died, I made it into Forbes magazine with the Nobel Prize winner, because it turns out that this is what people recognized as created community in our society. So that was quite an honor. And I realized this is another persistent story, right? Just keep sticking with your guns and do it. As long as people give you a platform, you kind of do it. So what happened from this show? So the first show, I realized at that point, I'm not the only one creative in science. It's kind of a lot of people. And so I'm going to show you a couple images here. So this is probably the most beautiful gonad you'll ever see. So Abby Dernberg is one of friends of mine now. So this is the C. elegans gonad, and you're looking at DNA in the germline of this gonad. One of the things about the art show has revealed is every person I'm going to show you that's doing art is a faculty member. And so that tells you that creativity and ability to do beautiful imaging also is a part of science. Irina Kolutev, these are C. elegans having sex on a batik scarf, right? You don't know what you're going to see, but wow, people are doing batiks in their free time, right? So James did this Andy Warhol. So here's his actual images, right? And he's doing an Andy Warhol kind of riff. I'm, again, always amazed what people show up to submit. These are more recent. So Dr. Varsha Singh is in the Indian Institute, unbelievable. He wants to communicate olfaction and C. elegans, right? This is in this Indian technique of this unbelievable, beautiful drawing, hand drawing. I'm just honored to even be able to see all this stuff all the time. Here's something that I do. So here's something I did with the worm and the embryo development here. And if you know this is the famous Moreau painting. If you've been in Barcelona or New York, you'll see this is Smithsonian. So that's my kind of take on, I kind of do that for fun, to kind of recreate old paintings with the worms. The other thing I do is have themes. And so in 2019, I said, I like Banksy. Let's try to figure out how can we do Banksy with model organisms. This ended up being submitted. So this was actually an actual graffiti. It's the world's largest model organism graffiti that I actually instilled in people. Philippe just got his PhD this year in Vienna. And this was easily a grand prize winner. And this was obviously painted over, but what happened from this event launched his whole side hustle job. So Philippe now does installations in science institutes all over the world, model organism, spray painting in the building. So a lot of the art show has launched a lot of people's science art, kind of passions doing other things. And so I have connections with all the people who have ever submitted into the show. A lot of them still in science, and a lot of them are actually amazingly faculty. And what's often said at the meeting now is if you don't enter the art show, it's real low chances of becoming a faculty member, right? So it's kind of like this funny thing, but it really created a lot of community within our society. And I'm continually asked, and it's always fun every year, what gets submitted. So when I became a faculty, I was able to get a lot of more opportunities. So I wanted to share a few of those things here. In my when I showed up, I was in fortunately a brand new building, and there was an empty wall here. And I said, hey, I walked up to the dean. At the time I was an assistant faculty. I didn't know there was a protocol. You have to talk to your chair, and your chair has to go up the dean. I actually just went to the dean directly and said, hey, I have this idea. And he gave me $15,000 out of the college budget. And I did this big installation. This got me in the art department. So a big installation in a building is kind of a big deal. That got the word of the art department saying, okay, we want her. This is really great. So this led to a lot of other opportunities. So now busloads of kids come through here as they go to our outreach program. And these are just representative images from things in our building just to introduce people to larger than life model organisms. From that, the art department contacted me, and the chancellor donated his own money, about $10,000, to get all these images framed and in the display cases in the Madison airport. So this then got a lot of press in the Chicago Tribune and NPR, and lots of stuff happened from that. Certainly there's lots of science artists now on campus. We kind of still judge this, and it became another competition, but it was the most popular show the airport had ever had. And there was a bowl that you can leave your notes. Most of the public was kind of shocked about what stem cells look like, how beautiful stem cells are. They're very curious. People are very curious about this, but I'd never had the opportunity to see it. So it was really nice. I sat at the airport for a while just watching people look at it. It was kind of really a cool experience to see it. And then, or just asking questions. So now I'm going to ask the audience, so you can unmute yourself if you like. I want you to guess what these things are. Does anyone know what it is? Scientists, you could kind of keep quiet, but I don't know who else is here, but do you know what this is? Anyone could say anything.
Drosophila egg.
It's a Drosophila egg, yes. This was on the front cover of the New York Times. So this is a fruit fly embryo, and these are the stripes of pattern of gene expression that actually know where the wings are actually placed and where you make the different body plan. This made it to the front cover of the New York Times partly at Easter because it looks like an Easter egg. So what you kind of recognize, that the science that kind of gets picked to go, it looks like something the public kind of knows already. And that's the beauty of the communication side of it. So we kind of pick things like that. That's how they kind of get picked. So does anyone know what this is? Anyone go hiking? Does anyone get attacked by a plant before? You probably have. You get like the sticky part of a leaf. So this is actually a Arabidopsis, a mustard plant. This is a trichome on the other end. It fends off defenses, but I think this looks like it's from Mars. So it's a very popular image because I was like, where is this from, right? But it's actually on a plant, right? Something very harmless. So there's that dichotomy between that. And then that cool science image that Tiny became this cool science image competition, which is a collaboration between ProMega, which is a biotech company in Madison, the art department, and the McPherson Eye Research Institute, of which I'm a faculty member. So they're very interested in vision and communication. You can find all of these things at the Vision Gallery here. And I'm going to share with you a couple winners here. So this is what the tongue looks like. So these are things that I've always wondered, what does my tongue cells look like? Well, they're all in these different kind of perpendicular arrangements of cells. I was kind of surprised by that one. Gold melting, and then you have a cerebellum. And this would be an image that maybe wouldn't make a journal article for a scientist, but it looks very
Ahna.
ethereal and painterly. Right?
Ahna,
Yes.
that's the hippocampus. It's not
Oh,
the
the
cerebellum.
hippocampus?
That's the
Okay.
hippocampus.
Well, that's
Sorry,
good.
being the neuroscientist here,
Well,
sending
that's
you
good to
chats.
know. I
had to-
remember in
had to
their
point
thing
that out
they
for
said
you.
it was this. They said it was the
No, the
cerebellum.
cerebellum is quite beautiful, though. It's quite beautiful.
It is very
though.
beautiful.
But-
Well,
but
thank you
that's
for
the
that
hippocamp-
correction.
But
Thank
that's
you
the
for that
hippocampus.
Thank you for that correction. I will correct my slides. So, then in 2018, we had a lot of chemists kind of win. This was the grand prize winner of tungsten, right? So sometimes these things kind of show up that are, and they kind of look the same color. We got the blood vessels of the brain, and then indium and cobalt. I mean, when you mix things together, you don't know what's going to happen. So that was kind of a very cool reveal in 2018. In 2019, Again, colors kind of switch. Things are always coming up. This is a cross section of a corn grain. This is a barnacle. And then this is iron ore and hematite. And what does this look? This was a grand prize winner. What does that look like to you?
Klimt.
It looks like Klimt.
Klimt's painting, yeah.
So it's like Klimt. Yeah. So those are another thing on the committee with scientists and artists, that those are actually, it looks very similar to a painting you've seen. So this one, it was a couple years ago. I want you to guess what it is. This is my most favorite one that's been submitted, partly for the obvious reasons. It looks like a very famous painting, but I want you to guess where in your body you think this is. Anyone? Not a guess? What does it look like to you? What painting? What artist? Alex?
Looks like Monet.
It looks like Monet. Yeah, it kind of looks like Monet.
I see Starry Night on this.
Yeah, Starry Night. So it actually is the blood vessels in the back of your eye. So here's some blood vessels here. It's kind of weird, right? So, but it kind of looks like kind of Monet-ish, Van Gogh-ish together. But this is the one when I talk to young kids that they go, my gosh, I have a Monet in my eyeball, right? So that's the part where the kids get really excited about, like, oh my gosh, this is really remarkable that there is this art there to discover in your own body, right? And that's where the interface with art is really great way to get kids into science, is to say, you can discover these beautiful art pieces in your body. Your body and organisms around the world is basically an art museum just left to uncover, right? So that's, and it always still drives me too. So that's really amazing images. So when I became a faculty member, I had students contact me. We heard you are very interested in art. And so an undergraduate came to my lab who had Chanel. She's a performance artist and costume designer. She still does this professionally. She's in New York City and also in Seoul. She wrote a Guggenheim Fellowship. If you're an artist, this is a very big deal. So she got a Guggenheim Fellowship as an undergraduate. She came to my lab and looked at the skin of the worm, the cuticle of the worm, and she made fabric that came from that experience in my lab. And she did a whole performance piece at the undergraduate research symposium. And so she did a whole process of what differentiation means. How does that do through development? And so that was a really initial experience that my lab saw that art and science is really important. There's kind of pushback from my grad students. They don't understand. Then they realize she knows how to use Illustrator and the art side of the things that my students have to learn. So they're constantly communicating with each other. And it was a really great experience. From that, I got my first Master of Fine Arts student. Angela was a former high school art teacher. She worked in museums and decided to go back to get her master's. So she did her MA in my lab. She came to the floor and worked with all the scientists. We had zebrafish and Drosophila and mouse, and my lab in C. elegans doing her project's called Translation, another science thing, translating the art in another way. And these are just, she's a photographer by trade, but kind of was into antiquing, making old and new. And so kind of went with kind of the young and old embryos of the zebrafish here. And she mounted them on sort of making science journal articles more into life and intuitive. So these they come out you at different heights and levels. So I really like this is on my floor outside my lab. And the great thing about these experiences, most science buildings have nothing on the wall or very little. And I said, well, why can't we just have all the artists collaborate with us and put all the science art on the wall? So that's my fantasy, to get the rest of my building done this way. And so these are other images. She took pigments and dumped it on E. coli. And she came up with this super innovative way, and I'll zoom up on that, to actually mount this. So it looks like a microscope slide. So the scientist, if you know what a microscope slide is, she's now mounting her art into the microscope slide. And these are just pigments that we got from our craft store, dumped it on E. coli, and it kind of grew up into these patterns, and she got really interested. There's nematodes crawling around in here, but think this kind of innovation of science, art, and communicating and going back and forth with each other. I'm going to show you, I have a couple movies in here about these projects. We always film this for disseminating it further. So this is the translation movie. And at the end, I want you to look at this cabinet of curiosity. She actually etched worm tracks on the outside of the box, and inside is a video of the worms. And this was our opening night, and 150 people that have never been in a science building came to this kind of art show. There's some music here, but there's no kind of narration. It's just kind of sound. But I want to get you a sense of what this installation looks like. Here's this cabinet of curiosity. There are the worms kind of crawling around in there. So that's the translation show, and I'm going to share with you. It's almost near the end of my talk. Is this genetic reflection, which is sort of my science, art, broader impacts with the National Science Foundation. So I was commissioned by the Biotech Center, which is adjacent to my department, to create a 40-foot piece. So this is a massive project for any artist to create a 40-foot piece that would fill in here. We have busloads of kids come to learn about DNA in here. So this was my only thing I was given. We do stem cells, the sequencing in this building, but they want, the teaching labs are down at the end here. So this was an enormous thing. And because Angela and I kind of worked together. So from this, I wrote an NSF grant with my science as well. My broader impacts is called, and decided, because Angela learned how to do that antiquing photographs. She learned how to silk screen onto mirrors, and I didn't show that piece of hers, but I got inspired by that. So I thought, oh, let's silk screen, stem cells and genomes onto mirrors, and we would have students but you would reflect the genome inside of you. So this is the concept. This is actually not a real photograph. It's a drawing by my brother, who's also a Syracuse grad. And so I put this in my grant, and I got a call from the National Science Foundation, and I neglected to put a budget for this art piece. So this is very expensive. And he says, if you put 50,000 in your grant, we'll fund your science. And what happened from that is they didn't know on campus that how to put money back in a grant. Usually grants get cut. And then what happened is they said, you do such amazing broader impacts, we want to fund your science. And they ended up giving me another 180,000 to support my science. So that was kind of like, what? It's if I won the jackpot. So they loved this. And then that became very unfeasible because this is very heavy and expensive. So I'll show you actually what happened is just like scientists, you have to plan out things like you would do in an artist. So it turned out we didn't have enough budget, so we had to reinforce the wall on the outside. So we had to map this out, what we wanted to do. And so it was lots of lab meetings and figuring this out, lots of test samples and printing this idea. We had 2 3D mock-ups like you would with like an architect would do. We had to go with building staff, and a lot of people were involved. If we actually had to do it, it would cost over like 50,000 to demolish the wall and do this in the building. So we had to come up with some other way to do it. And we always document with the videographer, which are students. So we have students that have another project, and we always teach them how to communicate science in different ways. So Matt was learning how to do that. And so always kind of mentoring the science communication. So here's Matt and Angela. So Angela learned, we came up with this idea of inverting, we're trying to teach about complementary DNA strands, and we had to figure out the right font and the right spacing, but there's a reflection idea to this. And this is what it ended up being. So it has this wood. It's not exactly what I wanted it to look, but it looks great, and it's better even in person. So these are different model organisms, ribosomal RNA, DNA that is there from every species that kind of we have on campus. And the function of this is that you look in and you see the reflection of yourself, and you see the DNA in there. So there's human down there as well. But what happens is students kind of see that the letters are the same and some and a little bit different. It looks better. And then the other thing is the light path. So what you're reading is the reflection. So we have the genetic reflections, and you can kind of see the words are actually reflected by the light. And we figured the exact angle, and we're also teaching optics, that light and light path is important. And this ended up winning. So Angela kind of won the citywide innovation in the arts for trying to communicate science. And here's another picture. It's a picture of my friend Alejandro took of me. You can kind of see the complementary DNA strands here that are reflected in there. So you really get a sense that you're part of this and that DNA is a part of you. So this is the genetic reflections idea kind of came up with. So there's another movie of this process, and also the teaching lab that's going to talk to you about the impacts that this has made on the students and them that come through there. This is a little low.
Public art is important in this building in particular because it gives our visitors a chance to see ideas that otherwise they might not be able to see.
So a lot of things in science you can't see visually with the naked eye. Like if you ask students to see DNA with the naked eye, they say no, but then here you are, you're literally walking by it. So take something abstract and makes it concrete. I run with Tom Zinnin the teaching lab. So we have K through 12 kids come in and we run workshops with them, specifically DNA based. We start them at the cell and we discuss that, that macromolecule and the shape of it and how it runs. And then as we walk down the hall, we talk about those letters, especially if you have like the little kids, they're like, wait a second, this is the 4 letters that we just talked about. And like some of them will like literally like stop and they'll read like, for 30 seconds. They'll like read out loud all the letters. And they're like, huh. So to make like that direct connection, the fact that they can interact with it so closely, I think is a really big deal with this art exhibit.
There's also cool things like, geez, if you don't have the light on, you can't really read the names of the model organisms. So they start getting the idea that lighting matters not only in art, but in things like microscopy and how we try to see things in the life sciences as well as in the arts.
So that's the piece. And one of the parts of the National Science Foundation Broader Impacts, you have to disseminate. So you can't kind of keep it on your campus. So this came from this coloring book, which is in my background. So my 2 talented undergrads, all of my undergrads are science art majors. So they come to me as genetics majors, and they're kind of going through the art thing and they want a project. So I said, hey, why don't we come up with a coloring book that is a tangible thing, and we can kind of, the goal here also was to raise money to support underrepresented students and women in STEM. And so my 2 talented undergrads, Alif and Caitlin, Alif wants to go to medical school and Caitlin wants to work for the CDC or WHO. So they spent a whole year, and I'm also in life science communication, learning how to do this whole process and publishing in Adobe InDesign and drawing this. And I wanted to take you through a couple images. It's an ABC book of those model organisms, of that parts of that piece. So it starts with Arabidopsis. And you can see the different techniques. This is actually Caitlin and this is actually Alif. You can kind of see their techniques. F is for fruit fly, N is for nucleus. Alif has this little kind of motif here. So it's kind of abstract there. Coronavirus came out in the middle of this right about published. So we had to change our V into virus in there. And so that's a popular page with the kids. And most importantly, we get lots of our little fans come in This was one of our first fans from a daughter of a geneticist that kind of sent this in. So this is always really what we're all doing this for, right? The kids, right? So a lot of people ask me, I'm going to end my talk here. Is this just me, right? Is it just Ahna? I revealed it's not just me, it's a lot of people. Someone sent me this data that I think you'll find interesting, is that it's from this paper from Roots and Burstein. It says scientists tend to have hobbies in music and photography. So red is scientists, blue is the general population. So that's interesting. How many of you are kind of into music and photography if you're a scientist? I think a lot of people kind of are. So this is kind of, it's interesting data, but it was never kind of quantified. And then the paper also goes on to say Nobel winners actually have shifted this a little bit, is that they have more hobbies in the arts and crafts, music and writing than the average scientist, right? So if you're kind of doing arts, craft, music and writing, guess what? Your chances of getting a Nobel just increased, right? So yeah, and then at the end of this paper it says, compared to general scientists, award-winning scientists, again, there's a bias group here, National Academy, Royal Society. If you're doing arts, right, you're really going to, that is where it's basically tapping into this creativity idea. So lots of questions get asked of me, and this was another one. How do you use art in your science? Why does it make you more successful? So this is a picture of a genome assembly, right? I'm just generically, that may not mean that to you. If you're an artist, it doesn't make any, but this is a scientist who decided, I'm going to arrange it in this way. This is that scientist's view. I would have arranged it totally differently. You would arrange it in a different way too. So that means is that these kind of aspects of science, so this is from Cyril Smith, as a metallurgist, said, any aspects of a large and complicated problem arise from factors you can't measure, right? It's for these, the artist's approach, uncertain over it seems it conveys more meaning. The way you see science and communicate it is really important piece of it. And if you can't communicate it in a clear way, then no one's going to understand it, right? And this is hard to understand for some groups. It's easy to understand for some people. But for even the public, you can appreciate how beautiful a genome assembly is, even if you don't know what it is. But I could do it totally differently, right? But people cluster things with color naturally. And those things are important for matching things up and pairing things, which are really important in science. So what have I learned from all of this is definitely science and art can inspire and engage the public really easily. Art is really able to communicate very complex ideas about the retina, for example, into a wide variety of audience. It's really about attracting it. And then working with artists really inspires new ideas in the lab and also creates new things that you would expect to have, new types of fabric, right? And in general, for my own self, I thought I was kind of alone in this when I started this story with you, is that I realized that scientists are generally very creative people and have a lot of cool hobbies that people don't talk about, right? And in that sense, if you're an educator in education, is that really encouraging students to pursue science and art. It's really necessary for the innovation in science. And for me, going to Syracuse and being able to be a biologist and to minor in ceramics and to go do that, I mean, Syracuse is a big art school. There's amazing opportunities there. And now it's a massively big science school. It was a very fortuitous for me to end up there, to have that environment to allow me to do those things, because I wouldn't be myself if I didn't end up at Syracuse with those amazing people that came into my life. So I hate math, but this is a Venn diagram that kind of describes me to a T, is I want you to think about where you find success in your own life. So what are your Venn diagrams, right? I found success in kind of the overlap there. It could be music for you. It could be architecture. It could be, whatever you have there, but you can find success doing interdisciplinary things. And I think we all are very interdisciplinary. It's just the education system makes it less interdisciplinary, but we now know that these interdisciplinary programs are very, very important. And I found success there. And I want to leave you with this. And I think as someone who always feels out of place, and if you're a student who feels out of place or a woman in STEM that feels out of place, is that you know, being able to see things differently than other people doesn't make you crazy. It really makes you valuable to the whole science, the public, the art world, everything. It really makes you valuable. And with that, I will take questions. I'm sorry I went over a little bit. So
Have our first question from Sandra. Do you want to ask it yourself, Sandra?
Sorry, unmute. Am I unmuted?
Yep.
Yeah, I don't have a question. I'll just read what I wrote. You're like, I'm so glad you persisted. I'm sorry. I can put my, I know it's hard with the video. I'm just sort of sitting in the, sitting here. I mean, what an amazing career you've had. It's so inspirational. And I know how difficult it clearly was for you to get where you were. And I want to shout out to all the mentors that, you know, just pushed you in the right direction that you mentioned, and probably those who didn't. And, you know, I'm like, I want my son to go to Madison.
Well, thank you. Thank you, Sandra, for that. Appreciate it.
That's all.
Thank you. Anyone else? You can ask me anything.
Oh, I don't want to do that.
Can I just
Go ahead, Melissa.
Oh yeah, sorry. I don't know how to use the Zoom hand, and I've been teaching online for like a year now, and somehow I don't ever use that. No, I was just curious how you if you incorporated art in your teaching, like in your classrooms, but also with microscopy. I look at chironomid head caps, those little larval midges under the microscope. And the person who trained me was an artist, and I could not see these morphological differences at all. I'm a writer, I'm not an artist. And so, you know, and until he drew them for me and actually drew them and started using shading to show me stuff, that's how I learned morphology, was from an artist. So I'm curious. Yeah.
So I teach a course right now, and I teach a genomics course, but the way I teach it is I teach the techniques of genomics, like sequencing and RNA sequencing and chemical genome, proteomics, all this stuff, the techniques of it, but I do it under the veil of science communication and visualization. So students come to my class and they think they're like, oh, I got to memorize all this science stuff, but they have to actually learn how to communicate it and synthesize their ideas. So they're doing it right now, and we do that in their talks. So they actually, how do you communicate that complex idea, or how do you explain RNA-seq and knocking out mutants? And you have to colorize, you know, you have to make sure people get it because they only have 30 seconds. And they're kind of like, they end up, and I also, because I'm a horrible writer, I'm a dyslexic, and I always work at it. I'm better than I was when I was the undergrad and certainly grad student. But I've learned to write better by seeing. And so I teach that to them, and I have them work on their talk. We did it 3 weeks into class. Okay, visualize what your project's about. And they go, what? I don't know how to do this, right? I said, well, let's just start. And I then have them write from their visualization. So this is actually how I write grants. I put all my science in a talk, and each slide is either a paragraph or a couple sentences. So and I know in a classroom, you have all these different learning styles. I know, because I was a dyslexic myself, is that I'm not good at listening. I'm hard to listen. I have to see something first, right? So I find those students that are kind of left behind in science because they're kind of the dyslexics that they really have to piece the words with the picture together. So I do that. And then what happens is they can see their gaps in their writing from trying to visualize that, and we go back and forth. So we do all the art of the science first, and then they write the writing, and I teach them how to write grants. They write a specific aims page, one page. I go, oh, it's kind of easy. We write one page thing. I'm like, well, welcome to my life. It's like I spend months on writing one page. So they write it, and they realize they're like, what am I missing? I said, well, why don't you visualize it? And they can actually find problems in their writing by seeing it. And then they go back to the writing, we go back and forth. So that's, Melissa, how I do it. And I don't lecture at all. It's all I have the students lecture and teach and use the visualizations to do that. And what actually comes out of it is a lot of students realize this whole side of science and communication, there's a job for them there. And so I had a science artist in my class yesterday who works in textbooks and works in medical illustrator. And I have at least 2 to 3 students per year go on to medical illustration programs. After that, some work for, they want to work at Smithsonian or National Geographic. These are parts of jobs that students may have never recognized, and it comes out of my class. Other students, I teach them how to make websites, also visual, something visual with writing. They then create their own companies, and they're doing like logo design for biotech companies. So that's how I do it, because it keeps me kind of sane, because I can teach them that color theory and contrast and light and, you know, all of those things that I just is inherent in me when I communicate, that I try to teach the students, because the students see what every other faculty is. It's not all text on a slide. You have to put pictures together, because in a classroom, it is very diverse. You can look all the same, but the learning styles are all different. And people don't forget about those invisible disabilities in your class. But I know that because I was that student, and I had a professor that I did project-based learning, and I drew out everything. I found all of my original papers. I drew everything out, and the writing was better for me. So it's just, and I like to teach them that technique, because, and the writing center on campus got a hold of it, that I was teaching writing through visualization. And the kids, and I never look what their GPA is coming in. The students that are like C's and B's are phenomenal at asking questions in science and also answering them in different ways. So those are the people who come to me and say, I thought this wasn't for me, and now science is for me. That's, you know, this is anecdotal, but this is kind of a cool result of allowing to assess if you assess students in different ways, with the visual and the writing and the verbal and the oral, you'll find students a place for a student in science across the board, right? So it's kind of long-winded, but that's what I do.
So Ahna, someone in the audience has asked if there's a book of some of the art presentations or galleries.
Of my work, or?
Yeah.
I mean, the only thing we have is basically, I have everything on my website, but that you could go look at, but then the only book is this coloring book that we have so far. So that's on Amazon or Blurb, you can find it.
Okay, another new avenue to go down.
Yep.
Coffee table books.
Anyone else have a question?
Yeah, I have a question.
Yep.
In a comment, I thought your talk was wonderful, and there's so many wonderful insights in what you talk about. But one of the things that's kind of missing in your talk, which I think it's important to recognize, is when you talk about children having the ability to do the things that, you know, that come naturally, to be creative and things like that. A lot of that is beaten out of children, you know, by our socialization system, et cetera. Yep. And I find it really remarkable that you really, what you're talking about is overcoming fear, right? And you touched on that with the setback in your own pursuit, you know, when the person said you're too creative. And and I find there's an interesting model coming up relative to that, is that creativity can be dangerous to some people. And one of the things that's happening in our languages is, as we're dealing with the COVID-19 situation and the immunology and the vaccines, that we now are really fearful of the variants, right? And the variants, for me, they represent a certain kind of dangerous creativity, right? So I think there might be a really big societal model here, because the arts are based in subtle innovation that could be much like the modeling of the mutation of this virus. And I don't think, obviously we're afraid of these mutations and variants because they could kill us, right? So I think it does present a situation for talking about creativity.
Yeah, I agree with you, Tom. I mean, I think general people are afraid of different things that are different, right? I think we're different of each other. I mean, look at the racial problems. We're different of people who look different than us, right? And there's a fear there. The fear is just unfounded because it's a stereotype, right? Like you have a stereotype that this is going to cause harm, or these people are going to cause harm to me, right? But the fear is, if you take a look or you have dinner with someone different than you can kind of reveal there's more to it, right? And so it's taking that one step and seeing that there's nothing to be fearful of, but you can be fearful, but you have to respect differences, right? So, and that's really just an important part of that. But yeah, I mean, I could go talk a lot about that, Tom, about this kind of ideas. You come into it. I came from a household of just boundless, unusual artistic creativity into an education system that told me scientists need to be this certain way, right? Or scientists are mostly male and maybe you'll meet one or two women. And I mean, that was my experience. And so I didn't know, the fear was, I was fearful that it was an environment that I wasn't able to succeed because I have to mold to what the path is. I have to be judged to tenure. I don't get judged on any of my art stuff for, my promotion. This is the crazy part of the whole thing, right? None of this work means anything to the scientist in terms of promotion. In that sense, I've had a lot of pressure in my career, right? But to the artist, this is a remarkable thing that I've done, because they kept calling me up and said, why don't you have tenure yet? So this is that's the kind of thing, is it's fearful, is that what I'm doing is so different. There's no way to judge it, right? Because it's so out of the box. But luckily I persisted. And I just said, well, I'm still going to do this work because it's important to me. And it's also now all science communication, the field was there, but not as how big it is now. And now it's very accepted that this, people are studying this, and this is a whole field. So it's I was well ahead of my time, but I've dealt with a lot of setbacks in my own professional career because it's still not judged as value to a university doing interdisciplinary work. There's no way to judge me, right? And that's why people are feared to do these kinds of things, because there's no way to go through the system, because you're spending too much time doing art and doing this at public engagement. But when you reflect on what I'm doing is really getting the public to be involved in the science process together as a whole, this is where our money comes from. I knew that as a kid, a poor kid. My parents are spending their little money and sending it to federal funds for science, but I never see it, right? So I knew it was all intertwined, but I kept, but it's fear. It's fear because it's new and different, and change takes time. I'm happy to see. I know Heidi is a good colleague of mine, and she also is a science artist. There's lots of people interested in science art here, and a lot of students now love science art. So I'm happy to be part of the initial trailblazers to kind of go through the system and figure out, how do I balance this? And you see success happen regardless of all of these things. And then people aren't so fearful. But again, I still get people that don't like what I'm doing because it's not that's not how they judge science, right? But that's reality, right? So, but I think I'm more complex than that. And I can't change myself, right? So I'm going to stick with my guns and just keep plugging away. I always have 4 or 5 jobs lined up on the side for fear that someone's going to boot me out of this business, because I'm always trying to, I kind of always feel I have to have a side hustle because I'm worried that I'm not going to be accepted where I am, right? But I think a lot of people do that. A lot of people are fearful that this is not where they are. A lot of people go to medical school and end up, oh my gosh, what am I doing here? I don't like this. This is not what I don't want to do this. And they do something else, or they go be a cook or chef. But those are great things. They find themselves, right? But I agree with you, a lot of those things, but there's a lot of creative people in this world, and a lot of them are all here, listening to me today. So any other questions?
Yeah, we have another question here in the chat for you, Ahna. So to what extent has art making helped you trust your instincts as a scientist? Has your artistic side made you a more intuitive scientist?
I think for me is I see things before I even know what it, I kind of know things before I kind of can write it down and communicate it in that way that scientists can do it. So I kind of see it. And John White said to me, I have golden eyes. He would say that to people. I have golden hands and eyes. I kind of know I'm also the way I actually think. I'm not like the usual dyslexic that changes letters around in words. I actually I put the last word of the sentence sometimes second. So I'll do but my thought is actually that way. So I actually know the answer, but I don't know how to get there. It's weird because it hits me first. It's like, it's very intuitive to me when I see things. So I kind of look at there and I get excited and I kind of read and I kind of reinforce my ideas and I keep going there. So, but it helps me to draw it. It helps me to see it. And then I'm all in. Then I've convinced myself I'm all in on the idea. So you could see that story on Monday if you're interested to show you this. It's kind of a very similar kind of story to this. It's kind of a long path of this intuitive, like, oh my gosh, this is it. And then we went all in and sure enough, it is. So yeah, I mean, a lot of that, I think a lot of scientists will say you're kind of know because you kind of feel it. But my head processes things in a different way. And I know the answer, but I don't know how to get there. I don't know the experiments to get there. Sometimes I don't know how to get there, but I know it's going to end up, and eventually it ends up right. So, but it's a good question. Anyone else?
There's just a few comments in the chat, but I don't think we had any other questions. If anybody else has a final question.
Final question. Alex, you look like you want to say something.
I mean, I don't think I have a specific question, but I will say that I thought the talk was absolutely incredible.
Thank you.
I feel like I've been thinking a lot about ways to incorporate more art into the kind of science that I do. And I don't know, this just was a really inspiring talk. It made me kind of be like, yeah, I think that's what I'm going to spend this afternoon doing, is thinking about ways to do that more. So,
well, that's great to hear.
Yeah, thanks so much.
Welcome to my class. I kind of call them people. Yeah.
It works, it's fine.
When you leave, I see people smiling. I was like, okay, there's a person, but I like to call on people. Anyone else?
Hello.
Yes.
Can you hear me? I'm from India.
Yep.
I'm calling.
Hi,
from
how are you? Oh, wow. Hi.
I mean, amazing chapter that you have covered for us today, fusing genetic reflections with your artistic fervor. And many times I thought it was like snorkeling under the sea, which I did in the Andamans, but unfortunately it turned out to be something very different. I was wondering, are there many scientists, artists like you in the United States or in the world? Do you have conferences and do you meet often?
We started during Zoom. There's a Science Artists Unite. Actually, we've got 2 people coming, Zooming in from India too as well. But we kind of started talking over Zoom to connect that way. But yeah, like Alan Drummond at University of Chicago does these models of like praying manti. He kind of does shows, and then Beata Merswa. There are a lot of worm people actually. C. elegans people that have done it, but I've met a lot of people over time. But we started talking. I met a soil scientist that makes pallets from actual. She's a geologist, soil scientist, and geologist who makes pallets for artists from the soil that she harvests. I think she's in Wyoming. She's I find them on Instagram too. So these amazing people, it's their side hustle, and they start these companies and they kind of sell stuff. My goal is I've been slowly finding, I just started a new company to try to highlight science artists and sell it. And so I want to be like the new Saatchi website for up and coming artists. So I'm going to do that for science art. So I just started it. It's called Skopology. So I'm going to kind of do this to try to elevate it more, with the goal that the public sees the beauty of science and to include science art in their everyday life, to understand that beauty is in science and your everyday world kind of thing. So that's kind of the goal. But if you Google science art, there's a whole kind of underbelly community. There's kind of a Twitter groups and stuff like that. And I love promoting and highlighting them and seeing what everyone's doing, because it's nice to see that they're doing. And a lot of the young students tell me, I'm here because I saw you doing what you're doing. And I think I'm one of the older kind of full professors that are kind of still doing this thing. I kind of am a trailblazer, and I think and they saw me do this, and they were doing it in the quiet, and now they're doing it loudly out on social media and doing it. And so that's really awesome to see. So I'd say go to social media and find us, and you can find really cool science artists. Instagram is a great place for art.
Yeah, I think so.
And so, and I found ceramicists that are doing, huge installations that I'm connecting with. And so, yeah, my goal now, I think later in life, is to try to promote more science art through my new website and company, to try to do that kind of stuff for that reason, but mostly for outreach purposes. So yeah, thank you for staying awake and listening to my talk. I appreciate it.
No, it's only 9:30 in the evening, yeah.
Yeah.
And I enjoyed it. And
Thank
you
you.
answered many of my questions, because I always thought, why are children so inhibited in attacking science, concepts, and they're so frightened of science, and there's a kind of a phobia instilled in them from childhood. But what I noticed in you were very determined that you will do what you wish to do. That's very important.
I think my dad's like that. Like, remember what my dad said, it's just kind of, there's two assholes on every campus. He's kind of like, you do what you do. You have to be you, right? Like the Picasso state, you have to be yourself, right? So it's kind of like my dad had that gregarious attitude, and I kind of, obviously I inherited it. So I kind of just, I don't really care what people think. I'm just going to do it.
Yes.
And so it's me. And I think that's hard to train people to do that, but they're there, and you can kind of get confidence in other students to just go for it, right? But confidence is really hard to learn unless you have professors that really build that confidence. So, and I had amazing faculty at Syracuse that built my confidence. So certainly I didn't know I belonged, like all the way, my junior and senior year at Syracuse finally solidified. I'm going to share on Monday some of my papers that I got back from my professors. So you can kind of see why I'm still in science. Yeah.
I'll get the link. Perhaps I'll.
Yeah, you should come. Thank you for your questions.
Thank you. It's been so wonderful, even seeing you and listening to you above all.
Thank
what
you.
a mind you have. What a mind. God's gift.
Thank you. So, any last questions, comments? Well, thank you very much. I'm honored to be here and give this talk, and I appreciate you spending time with me today. See you.
So to finish things up, WISE, the Norma Slepecky Committee, and our sponsors, Humanities Centre and Biology, we all hope that you really enjoyed yourself today. I know that I had a wonderful time. So thank you, Ahna, for sharing your journey with us. And please thank her for a wonderful presentation. And also congratulations once more to our remarkable undergraduate researchers.
Congratulations.
So thank you and congratulations.